Hello, everybody. Today we are going to first start the first chapter of 12th grade biology, that is sexual reproduction in flowering plants. If we are going through the first paragraph, that is the paragraph given as the introduction, there are very few points that is vital in terms of exam point of view. And here it just talks about flowers in general, it's like flowers, how they are essential in our lives, how flowers they are related with beauty and all, and how we are using them for our own selfishness, and that flowers they engage in sexual reproduction. That means they play a part in sexual reproduction. And there's a word, what, oh, aesthetic, it comes later, sorry. So this first paragraph, they're like, we enjoy the smell and beauty of flowers, and they come in rich colors. So myriads means multitude, and next inflorescence, it means how flowers, the arrangement of flowers in a stalk, and morphology. So I have typed like this. No, that's actually, I just typed into this PDF right now. It's not given in the text for easy understanding, so it helps to for me to easily teach you all. I have texted some meanings, so it will help me teach you. It's not given in the text. And finally, how we humans use them for our own selfishness, and the different inflorescence. And at this chapter, we can easily understand the morphology. That means the study of structure and form of plants and animals. Also, the processes of sexual reproduction. Okay, so a flower, a fascinating organ of angiosperms. This paragraph is also not important in an exam point of view. Angiosperms means they have flowers, and their seeds they have an outer covering carpet, and also they have vascular tissues, and they comprise a majority of the plant type, that is angiosperms. So these humans they have a connection with flowers, and they can be our objects of aesthetic. Aesthetic means nothing but it's concerned or related with beauty, how we appreciate beauty, that's aesthetic. Ornamental, social, religions and cultural value, also they have been able to signify important feelings like love, affection, happiness, grief and mourning. And also, if to the point of view of a biologist, they are morphological and embryological markers and the sites of sexual reproduction. So, if we, in the point of view of a biologist, for him or her, the structure and the embryo, the structure also, the properties of the embryo, they are very crucial. They consider it very eagerly, and also these flowers, they are sites of sexual reproduction. And next, pre-fertilization structure, I will use this little intro, that's also not important, but starting this portion is our important portion. So pre-fertilization structure and events. Okay, so if you're seeing an angiosperm, before even the flower it has taken its form, we already know that the plant it is going to bear a flower. And several hormonal and structural changes are initiated which lead to the differentiation and further development of the floral primordium. So hormonal changes and structural changes they occur, and also differentiation of cells and also floral primordium. I have texted the floral primordium. It is been a bud. It develops into a flower at the end of a stem. Inflorescences are formed which bear the floral buds and then flowers. In a flower, the male and female reproductive structures, the androecium and gynoecium, differentiate and develop. Next, there is a word which is whole. So, whole means concentric shapes, concentric spherical shapes. That's what we call whole. So, whole of stamens, androecium, it means a stamen part. See, what you say, anther and the filament, all of them present in the flower. They are arranged in shape of a whole, and we call it androecium. And also gynoecium, the female reproductive organs, they are also arranged in a whole manner. Whole manner. Actually, exactly, it's not present as a whole, but it's more accurate in terms of the male reproductive organ, the stamens to be considered as a whole. So just forget about gynoecium being whole, but stamens is surely a whole. It shows this type of circular or spherical arrangement. And next, coming to this statement, microsporangium and pollen rings, they are very important. Also, this, as you all know, all diagrams are important, and surely these two might be also important. Let me change the ink color to blue. So, um, wait a minute. I'll be marking the important points in this paragraph. So the important points are the two parts of a typical stamen, the long and slender stalk called filament, the terminal generally bilobed structure called the anther. If you want, you can also underline along with me in your textbook. So if you are considering this stamen, it is also the male reproductive organ. Now, as you see, it's very long and also slender. Slender means a skinny shape, and we call this structure only filament. It's stalk-like structure and also it is slender, meaning skinny. It's better to write slender instead of skinny in your exam paper. So please only write slender, not skinny. This one slender. If if you are to describe this thing, and then the next important portion it comes in the next paragraph. Okay, before that, this stamen it is divided into filament and anther. So filament I just described right now. Anther. Terminal means its central or main part. Terminal top region it is present. It's bilobed. Two lobes are there. It is bilobed structure. This one anther. It's bilobed. Two lobes are there, so we call it bilobed. And next, in the next paragraph, to underline this part. Also these keywords are written in bold, so it's easier to navigate the important portion through these keywords written in bold, texted in bold in this textbook. So the next important portion, a typical angiosperm, anther it is bilobed with each lobe having two teeth. You see, a typical angiosperm anther, it's bilobed. As I have already mentioned, it has two lobes, so it is bilobed, and they have two theca. So each lobe has two theca. This is one lobe, and also this is one lobe. Two theca means this area. This area we call it theca. So if we are considering this as one lobe and this as one lobe, it has two thecas. Here one theca, here one theca, here one theca, and also here one theca. I will use another pen. So once more, this anther suppose we horizontally cut it and we get a transection. This one transection we get, and obviously it will be fused with the two lobes. And we divide the two lobes like this, and next the theca, it is the area. It's an area of the lobe. This is one theca, this is another theca. Also the same goes for here. Understood? And since they have two theca, since each lobe they have two theca, it's called dithecas. A longitudinal groove runs lengthwise, separating the theca. It might be a bit confusing to understand which longitudinal groove. So this one, this one longitudinal groove. What does it do? It separates it. It separates the theca. It separates both the lobes and the theca. This longitudinal groove also it runs lengthwise. It extends up to here. The longitudinal groove. Now coming to the very next important portion. The beloved nature of anther is very distinct in transverse section of anther. The anther is a four-sided tetragonal structure consisting of four microsporangia located at the corners, two in each lobe. The beloved structure of anther is very distinct in the transverse section of the anther. The beloved structure. So consider the anther. The beloved structure of the anther. It is very distinct in the transverse section of anther. So I have talked about the transverse section of anther. You can easily distinct and say it is a very distinct structure. You can easily figure out with the transaction that it is a part of an anther. So anther it is four sided. As you see, there are like four sides. These four sides, and it gives a tetragonal shape. Tetragonal shape means four sides, and also it consists of you see. So this consists of four microsporangia. Here one, here one, here one, here one. So this outer layers and consisting of this undifferentiated cells, it constitutes a microsporangia. This part, this outer layers, it will be described further in detail just now. For that, this part on a total, we call it a microsporangia along with the layers and also inside it undifferentiated cells or sporogenous tissues, but it will be described later just now. And the microsporangia located at the corners. So, as there are two thecas in a single lobe, there are also two microsporangia in each lobe. Next, the very next important portion. The microsporangia develop further and become pollen sacs, and they extend longitudinally all along, all through the length of an anther, and are packed with pollen grains. So what happens to the microsporangia? The microsporangia they developed further and they become pollen sacs. Inside it, pollen grains are present. So this microsporangia, it won't stay still like it, right? It will soon develop. It will soon develop, and what does it become? It later becomes pollen sacs. They extend longitudinally all through the length of the anther and are packed with pollen grains. You can see pollen grains. This microsporangia soon, very soon they mature, and then they later they become pollen sacs, and they extend all along the length of the anther, and they become fully packed with pollen grains. Next, structure of microsporangium. A typical microsporangium appears near near circular in outline. It is generally surrounded by four wall layers: the epidermis, endothelium, middle layers, and the tapetum. The outer three wall layers perform the function of protection and help in the dehiscence of anther to release the pollen. The innermost wall layer is the tapetum. It nourishes the developing pollen grains. Cells of the tapetum cells of the tapetum oh no, never mind. Cells of the tapetum possess then cytoplasm and generally have more than one nucleus. Okay, now I will explain this paragraph. So, typical microsporangium is circular in outline. No, this is not a perfect circle, but in using a mouse, using a computer mouse, only this type of circle is good for now. Also, I can't find shapes. So can you adjust with this one? This as a circle. Please consider it as a perfect circle. Okay, a perfect circle, microsporangia. This is the outline, and it appears near circular. It's not a perfect circle. It appears near circular, and also it is generally surrounded by four layers. As I have told you, four wall layers are present. Okay, this portion, four wall layers are present. I think it's better to use different color or it's easy to get easily confused. Oh god, I think I need to erase everything once more for the sake of your understanding. Okay, it might look like a rainbow. First layer, second layer, one more layer needed. First layer, second layer. Okay. Okay, so this is the first layer. So four layers a microsporangium has. This is the first layer. What's the name of the first layer? This layer. So the name of the outermost layer, it is the epidermis. Epidermis, this is the outermost layer, and later on, innermost layer, this is the endothecium. Endothecium is the next layer, and this is the middle layer. And after that, uh, this there is a set of tissues inside this circle. Inside this circle, a set of tissues are there, and before surrounding that, a set of another tissues are there, and that is called the tapetum. So what does the tapetum do? So the tapetum the tapetum, it nourishes the developing pollen grains. So inside this layer, there are developing pollen grains. So this layer, tapetum, this tapetum, it helps in nourishing these pollen grains inside it, developing pollen grains. And all other tissues, middle layer, endothelium and epidermis, they help in dehiscence and protection of anther. So they have protection function. So what do you mean by the term dehiscence? So dehiscence means and the inside the anther a lot of pollen grains are present, and when they mature or when the time is right, then the anther it bursts out. So the process of this bursting out, we call it dehiscence, and it bursts out and pollen grains are released. So this bursting out to release pollen grains, that property is called dehiscence, and these three layers they help in protection and dehiscence. Okay, the next paragraph. Hmm. Oh, sorry, I just took the wrong path. No, no, nothing, nothing. Okay, when the anther is young, a group of compactly arranged homogeneous cells called the sporogenous tissue occupies the center of each microsporangium, and as microsporogenesis, as the anther develops, the cells of sporogenous tissue undergo meiotic division to form microspore tetrads. Okay, when the anther it is young, so inside there is a group of compactly arranged homogeneous cells called the sporogenous tissue. Wait a minute. I will show the sporogenous tissue. Now there are a lot of cells like this within the tapetum. This region is full of undifferentiated cells. And these undifferentiated cells, we call it sporogenous tissue. So when the anther is young, a group of compactly arranged homogeneous cells, sporogenous tissue, in the center of each microsporangium, they occupy the place. Okay, so that's what is the sporogenous tissue, as I have been mentioning earlier. Okay, next, microsporogenesis. So microsporogenesis, it is very important. Maybe there is a high chance that this term might be asked. What term? Microsporogenesis. As the anther develops, the cells of the sporogenous tissue undergo meiotic divisions to form microspore tetrads. So they suppose the anther it tries to achieve maturity at that time. What does the cells, the sporogenous tissue, they undergo meiosis, and then they form these microspore tetrads. Also, these microspore tetrads, it's immature. Please note it down. They are microspore tetrads. They are immature form. Okay, so these are cells of this sporogenous tissue, they undergo meiosis. Meiosis means, when group of cells or a cell, they are in diploid condition, reduction, division happens and they become haploid. And they form microspore tetrads. Sporogenous tissue undergo meiosis and form microspore tetrads. And okay, the next page number 6. Okay, these three diagrams are important. They can be asked for labeling and also maybe for drawing. But there is a high chance for labeling and to be described further. Based on this diagram, they will just maybe give this diagram and ask to describe further. What is this? What is that? And we have to write a whole paragraph. Maybe like that. As each cell of the sporogenous tissue is capable of giving rise to a microspore tetrad. The process, the process of formation of microspores from a pollen mother cell through meiosis is called microsporogenesis. The microspores, as they are formed, are arranged in a cluster of four cells. The microspore tetrad. As the anther mature and dehydrate, the microspores dissociate from each other and develop into pollen grains. Inside each microsporangium, several thousand of pollen grains, several of microspores of pollen are formed that are released with the dehiscence of anther. Okay, so this paragraph, as each cell of this porogenous tissue, it is capable of giving rise to a microspore tetrad. So this as porogenous tissue, like we are considering a single cell from this microsporogenous tissue, a single cell if we are considering, it is capable of giving a microspore tetrad like this one. The process of formation of microspore from pollen mother cell through meiosis, it is called microsporogenesis. And microsporogenesis, they have like two definitions. One where this porogenous tissue they undergo meiosis to form microspore tetrads, or you can like the pollen mother cell when it undergoes meiosis to undergo to finally You divide to finally form microspores, microsporogenesis. Either way we can write. The microspores as they are formed are arranged in a cluster of four cells. Okay, so microspores, these microspores they are arranged in a group of four microspore tetrads. And these either it mature and dehydrates, the microspore they dissociate. They extend from each other, they separate, they farther away from each other, the microspores, and finally they develop into pollen grains. And inside each microsporangium, several thousands of microspores or pollen grains are formed and they are released with the dehiscence of anther. Okay, so what's a pollen grain? So pollen grain are basically male gametophytes. So gametophytes in angiosperms, they are haploid in condition. So gametophytes, what are their function? Gametophytes, they release gametes. So pollen grains, they release male gametes. Next. Pollen grains are generally spherical, measuring about 25 to 50 micrometers in diameter. It has prominent two-layered wall. The hard outer layer called the exine. It is made up of sporopollenin, which is one of the most resistant organic material known. It can withstand high temperatures and strong acids and alkali. No enzyme that decreases sporopollenin is so far known. Pollen grain exine has prominent apertures called germ pores where sporopollenin is absent. Pollen grains are preserved as fossils because of the presence of sporopollenin. The exine exhibits a fascinating array of patterns and designs. Pollen grains they are generally spherical, measuring about 25 to 50 micrometers in diameters. Their measurement they are very small, around 25 to 50 micrometers in diameter. They are spherical. Pollen grains, their structure. And it has prominent two layers. It has prominent two layered wall. The hard outer layer called the exine. The hard outer layer it's called the exine. It is made up of sporopollenin. So sporopollenin it is very resistant material ever known to mankind. Like it can resist high temperatures, strong acids and alkalis and no organic naturally occurring organic enzymes can degrade sporopollen. And also it pollen grain exine, we are talking about exine, it's very resistant. It has prominent apertures called germ layer. So germ layer it's a spot in the exine where the sporopollen it is not absent. It and through there pollen tube develops. Pollen grains are well preserved as fossils because of the presence of sporopollenin. The exine exhibits a fascinating array of patterns and designs. The inner wall of the pollen grain is called the intine. It is a thin, and continuous layer made up of cellulose. The inner wall of the pollen grain is called intine. It is a thin continuous layer made up of cellulose and pectin. The cytoplasm of the pollen grain is surrounded by a plasma membrane. When the pollen grain is matured, it contains two cells, the vegetative and generative cell. The vegetative cell is bigger, has abundant food reserve, and large irregular shaped nucleus. The generative cell is small and floats in the cytoplasm of the vegetative cell. It is spindle shaped with dense cytoplasm and a nucleus. In over 60% of angiosperms, pollen grains are shed at this two-celled stage. In the remaining species, the generative cell divides mitotically to give rise to two male gametes before pollen grains are shed. Three-celled stage. Okay, so the inner wall of these pollen grains, it's called intine. It is a thin and continuous layer. It is cellulose and pectin. Here extend it is made up of sporopollenin, whereas the intine it is made up of cellulose and pectin, and compared to exine, intine is rather slow. It is soft, thin and continuous, and it's made up of cellulose and pectin. The cytoplasm of pollen grain, it is surrounded by dense, no, not so dense, but plasma membrane and cytoplasm. The cytoplasm of a pollen grain, the cytoplasm, it is covered by a plasma membrane. When the pollen grain is matured, it contains two cells, the vegetative cell and the generative cell. This diagram, it is important. Vegetative and generative cell. The vegetative cell is bigger and has abundant food reserve. The vegetative cell, it is rather big compared to the generative cell, and it has food storage, food reserve, and also what does it have? It also has a large irregular shaped nucleus. Also, the generative cell, it is rather small and it floats in the cytoplasm. It is spindle shaped. It is spindle shaped. Also, it has dense cytoplasm. It has dense cytoplasm and nucleus. It's dense. Now, so a general statement, in over 60% of the angiosperms. Okay, so consider all the angiosperms be of a number 100, all the angiosperms, and out of that a portion of 60%, we are talking about 60 percentage of them, here the pollen grains, when this when this pollen grain, they are shed at two cell stage. When it has two cells, it will immediately shed. In the remaining species, 40 percentage, what happens? So the remaining species, the generative cell, it divides mitotically. Mitotically means no reduction division occurs, and also the same number of set of chromosomes, they are present at the final stage. And it mitotically divides the generative cell, and it gives rise to two male gametes before pollen grains are shed. So it gives rise to two male gametes. Before these pollen grains are shed, it gives rise to two male gametes through mitosis, and it occurs in three cell stage.